Our next speaker is Dr. Sharon Savage, and she'll be talking about the bench to bedside program. Okay, Sharon's over here. Good. Come on up, Sharon. She's going to be talking about telomeres and lifestyle and Li-Fraumeni syndrome outcomes. I think if you're not familiar with the bench to bedside program, there are a couple different components, and Sharon has been one of the successful competitors here for this program, so we'll have her talk to you a little bit about what her research has been going and where it's been going. So thank you.
There we go. Okay, great. Well, thank you all very much. It's wonderful as a clinician and as a scientist to be a part of what is a very exciting day and to see such a wonderful turnout. My job today is actually is going to focus on the science and on this bench to bedside program, which I'll talk about in a bit. But first, I want to start about how in the clinical genetics branch here at the National Cancer Institute, how we focus on rare diseases, biology, and how we use that information to inform not just rare diseases, but also the general population as well. So we do clinical studies here at the NIH Clinical Center where we do comprehensive clinical characterization, we do detailed family history studies, we collect biospecimens, and we work very closely with basic scientists to really understand the underlying biology so that we can figure out why these rare diseases are happening and then hopefully what we can do about them. And then this also has implications to the population at large simply because we're learning more about biology of rare and common diseases as well. So here in the clinical genetics branch, we focus on a multitude of rare syndromes, including the inherited bone marrow failure syndromes, a Dicer-1 disease, which is an autosomal dominant disorder caused by mutations in a key RNA processing gene, and high cancer risk, as well as multiple other studies of individuals at high genetic or acquired risk of cancer. One of those disorders that was first described here at the NCI in 1969 is called Li-Fraumeni syndrome, or LFS. It has autosomal dominant inheritance and is characterized by early onset cancers, typically a pattern within families of sarcomas, breast cancers, leukemias, brain cancers. And more than half of individuals with this disease develop more than one cancer. So just one cancer diagnosis is actually less common. Patients get multiple different types of malignancies. Germline mutations in the P53 or TP53 gene were linked to cause Li-Fraumeni syndrome in about 70% of families in 1990. P53 I bring up in that it is a real central gene in the cell cycle, in transcription, in activation. It's also important in metabolism. It's really this key gene, and loss of P53 in tumor cells is actually associated with malignancy. And these are patients who have a germline mutation in this key tumor suppressor gene. So many years ago in the branch, we started a study that really started because of international collaborations where we had opportunities to work with investigators who were doing a cancer screening trial. This was around 2007 or 2008. We had international colleagues who were studying the genotype-phenotype correlations in this disorder. And so we said, you know, if we need to get back in this game and really start looking at Li-Fraumeni syndrome in our own clinical center here. So we opened this study to accrual in 2011. It's a family-based study, which is how we do all of our studies at the clinical genetics branch, where we study the people with the disorder and those in the family without it so that we can understand what's part of having the disorder and what's part of being in that family only. So we are looking at biomarkers. We do detailed family histories. This is one pedigree spread out over multiple pages in our study team meeting. And we also have a separate component I don't have time to talk about on the psychological and social consequences of dealing with this high risk of cancer. We have a cancer screening study that's ongoing here at the NIH Clinical Center in these families as well. So in 2010, we hosted here at the NIH a workshop that, again, was prompted by inquiries and potential for collaborations from a couple years prior to that in multiple different areas, as I mentioned, cancer screening, biomarkers, cancer prevention, and Li-Fraumeni syndrome. So we had this meeting where we had 50 clinicians and scientists come from 4 continents, and we formed the Li-Fraumeni Syndrome Research Consortium, which is the first time that these outstanding investigators had really gotten together and started to pool our data, pool our resources, pool our brain power to advance this disorder. And then at the same time, in the same meeting, we had about 80 family members, people affected and unaffected with Li-Fraumeni syndrome, and we helped them really solidify what had been the beginnings of a family support group, which is now called the LFS Association. So this brings me now to the international research component, which brings us to Brazil. So part of our collaborations were looking at Li-Fraumeni syndrome in Brazil with Dr. Maria Isabella Schatz, who's at AC Camargo Cancer Center in Sao Paulo, and Dr. Patricia Prola, who's at the Clínica de Porto Alegre in Porto Alegre, Brazil. And so they had noted in their cancer genetics clinics kind of typical, what we would consider sort of typical LFS families with mutations in the P53 gene that are typically found in the DNA binding domain. And so this is really kind of what we would consider typical LFS associated mutations. So there definitely is a population of patients in Brazil with classic LFS. What was especially interesting in these populations, here we are in southern and southeastern Brazil, where in around the year 2000, there was a study that looked at 35 children with adrenal cortical cancer, so cancer of the adrenal gland, and it turns out that all of these kids, who were 35 kids from 26 families, had the exact same mutation, an arginine 337 histidine, in this different domain of P53, in this oligomerization domain. This was unusual because we had really what looked like a cluster of this cancer with this rare mutation. Subsequent studies by Dr. Schatz, Prola, and others showed that actually these carriers have a similar spectrum of LFS-related cancers that I mentioned earlier, but they seem to have later age of onset than a regular LFS patient and fewer cancers in the family. And again, we have a special population in southern and southeastern Brazil. Genetic studies actually showed that this mutation, specific mutation, was the result of a founder mutation. So that and what it was in the genetic analyses traced back to was an 18th century trader from Portugal, is the hypothesis, who came over for trading, and this trade route was actually this famous Tropeiro Trail that when they mapped out where their families with this founder mutation lived and where they had come from, it all sort of formed around this special population. And subsequent studies in Brazil with our colleagues have found actually this founder mutation is much more common than we expected, with about 0.3% of this specific population being carriers. Now that means that they're at increased risk of cancer. But what also was especially noticeable in Dr. Schatz's clinic was that these patients had a very youthful phenotype. So she had people who had not had cancer in these families with the mutation who looked 20, 30 years younger than you might expect them. And she also has noted that they had fewer than expected age-related diseases, such as osteoporosis, Alzheimer's, et cetera. So we developed this collaboration to follow up on our hypothesis through an NIH bench-to-bedside grant, which basically is just what it says. It's multi-institutional, and the idea is to take clinical observations, take them to the bedside, or actually start with basic science observations and take them to clinic. And we've kind of done a little bit of both here. I was especially interested in the idea of the aging findings with the P53 founder mutation, because another major part of my research is on a telomere biology disorder or a premature aging disorder called dyskeratosis congenita. And so we hypothesized that this mutation in P53 may have persisted in this population because it's associated with healthy aging, even though you have this high cancer risk. If you get through with the cancer, you actually can potentially live healthier and longer. Telomere shortening, I'll talk about those in a sec, is a consequence of the normal aging process. So we hypothesized that telomere biology could be connected with this specific mutation in P53. So telomeres are shown in yellow on the ends of these chromosomes here, are long DNA repeats in a protein complex at chromosome ends. And they shorten with each cell division as a consequence of the DNA replication machinery just can't replicate the last ends of DNA. So consequently, telomere shortening is a feature of normal aging. Here's age and telomere length and a range for healthy individuals. And we know there are some telomere biology diseases where patients have exceedingly short telomeres. That's dyskeratosis congenita. But we also know telomere length is associated with multiple diseases in the general population as well. And telomere length is especially tightly regulated by genes, by common polymorphisms, and by environmental exposures. So in an international exchange, we were able to work with Kelvin Andrade, who is a graduate student of Dr. Schatz, who came to the NCI about a year ago to do his graduate work. And he worked closely with Anna Best, who's in our biostatistics branch, and we measured telomere length in individuals with the founder mutation, individuals in our own NCI study who did not have the founder mutation but had a different P53 mutation, and also in their healthy, unaffected relatives. And what really jumped out at us in our very first analysis was that the patients from Brazil with the founder mutation did not appear to have the same age-related telomere shortening as we expected in the subjects from the US NCI cohort with mutations and from the controls from both countries. So this was really interesting to us, thinking, could there be a telomere connection with this aging phenotype in this mutation? So the next thing Kelvin and Anna did was look at did some additional statistical analyses to infer their telomere length at birth based on projections from their current telomere length, and also based on some kinship models where we looked at the additional relatedness within families. And again, it really looks like our patients who have the founder mutation, who don't appear to have telomere shortening over their lifespan as compared with the patients with different P53 mutations. And in fact, when we look at the estimated telomere length at birth and their age-related telomere shortening, we actually see complete separation in the Brazil founder mutations and the NCI non-founder mutation carriers. So this has really led us to, this is a study that's under review now, and I think if anything, it's actually generating more hypotheses. And what some of those hypotheses are is that this specific mutation in P53 is known to be detrimental because it does reduce P53 activity, and that reduction in P53 activity results in tumor suppression, so basically you can get more tumors and increase tumor risk. But what we found is that possibly in connection with other genetic factors that we haven't yet identified in this special population is that we see less age-related telomere shortening, what we think it looks like healthy aging, and a slightly milder potential phenotype. So this is really just the tip of the iceberg, but I put this forth as it's been a great collaboration in that we're working with multiple different institutions across countries and special populations, and this is really opening a whole new area of research in trying to understand connections between cancer risk, telomere biology, aging, and by working closely with these families, we're also developing cancer screening protocols, and hopefully we'll actually be able to do some of that precision medicine we've talked about throughout the day, in that we'll be able to better do genotype-phenotype correlations and figure out how to best do cancer screening and prevention in these very special populations. And so with that, I'll stop and just emphasize that this has really been a team effort with all of our families on both continents, across all of our institutions at the NCI and in Brazil. And I'd be happy to take questions if we have time. Thank you.
Is there a question for Dr. Savage? I don't see it, but Sharon, thank you very much. I think it really does reflect the need for, is there a question? Okay, question, please.
Yes, sir. Hi, I'm Grace. I'm with the National Alliance for Caregiving, and I want to thank you on such a fascinating presentation. I'm not a researcher, so this question may be silly, but I was thinking about the comments this morning about the applicability of research in the rare disease space to other fields. Mm-hmm. And especially the work here related to aging. I'm wondering, do you think there'll be findings that could be relevant to the dementia community?
To the dementia community specifically, so the way we approach everything in a lot of us that I think who are scientists studying rare diseases is we also have this sort of more common disease thinking in as well. And so what this example could actually lead to is more ideas connecting aging and the biology of aging, and then you could envision a connection to dementia or to other age-related diseases. This is really helping us understand the biology, which is really the first part.
Thank you very much.
Great.
Okay.
Thank you very much.